students, let us continue our discussion on biomolecules. Our next topic is enzymes. So what are enzymes? Enzymes are called biocatalysts. Okay, as any chemical reaction is accelerated by adding catalysts, so all our biological process, especially the digestion and all, they are all catalyzed by biological enzymes, and these are called biological catalysts called enzymes. So basically enzymes are mostly proteins. Okay, so you can see these are biological catalysts produced by living cells which catalyze the biological reactions in the living organism. Chemically, enzymes are naturally occurring simple or conjugate proteins. So that is the specialty of enzymes. They are naturally occurring. We cannot take them externally. Of course, we do take them at times. Suppose you have indigestion and all, we do take enzymes. But normally, in normal human beings or normal living beings, it is produced within the system itself. Some enzymes may be non-proteins also. Now you can understand life is a process of coordination of various chemical reactions in living organism. So you can understand our human body is a wonderful machine. The moment you start eating, your mouth starts its job, the teeth start chewing the food, then the tongue helps you in moving the food from one point to the other, and you see the salivary amylase, the enzyme is immediately secreted to start the digestion process. If any of these stop functioning, then there will be miscoordination. Okay, a process like digestion of food involves a sequence of reactions, and all these reactions occur in our body under very mild conditions with the help of certain biocatalysts called enzymes. Most of the enzymes are globular proteins, meaning they are soluble. They are very specific for a particular reaction. That is something very important. Now, as every lock has a particular key, every substrate in our body has a particular enzyme. So it is very much specific. You must have done that lock and key theory in the chapter surface chemistry. And a particular substrate, okay, they are named after the compounds of class of compounds upon which they work, or sometimes are the functions which they perform. So there are two ways enzymes are named. So one is the substrate on which they act or the function which they perform. Okay, so you can see there are some examples. Here you see maltose. When hydrolyzed, it gives you glucose. Maltose gives you glucose. Okay, and the enzyme which is used here is called maltase. Most of the enzymes, their names are ended with ase. So how it is named? It is named the way it is acting on the substrate. Another enzyme is invertase. When sucrose, that is our normal sugar, is hydrolyzed to give you glucose and fructose, okay, and you know the process is called inversion of sugar. Here the enzyme which is used, that is called invertase. All right, so here on what basis the enzyme has been named? It is named on the basis of its function which it is performing. So these are the two ways an enzyme can be named. There will be another enzyme which you will find by the name oxidoreductase. Okay, so as the name suggests, you can understand it helps in oxidation and reduction processes. So the enzyme which catalyzes the oxidation of one of substrate with simultaneous reduction of the other is named as oxidoreductase enzymes. So there are certain characteristics of these enzymes. Number one, they are highly specific in their action. Each enzyme can catalyze only a specific type of reaction. So one enzyme cannot do multiple functions. Okay, they are very particular for a particular set of reactions. They can speed up an uncatalyzed reaction to the extent of 10 million times. Let me tell you, if we don't have enzymes in our body, one meal would take 10 years to digest. 10 years. And you can understand, because of the enzymes we have, we are taking 3 to 4 meals a day. Whereas one meal would have taken 10 years to digest without the enzymes. The reactivity, they are active at moderate pH. So you can see normal pH is 7. So they act between 6 to 8. That means slightly lower towards the acidic side and slightly towards the alkaline side. and at moderate temperature of 310 Kelvin. Their actions can be controlled or even inhibited by certain organic or inorganic substances. That means if you want them not to work, then you can stop them by putting some external reagents, or even if you want to control their action, that means sometimes we feel too hungry, number of times we are eating, that may not be natural, so you can stop that process by taking some external, we call them medicines or some other processes. Even a small amount of enzyme can be highly efficient as they are regenerated during the catalytic activity. As you know, all catalysts, they are regenerated at the end of the reaction. So even the enzymes are regenerated after they perform their function. So with this we come to the end of discussion on enzymes. Our next topic is vitamins. So vitamins, they are organic compounds which are required in small amounts for the healthy growth and functioning of animal organism. Very few are synthesized by animals. So you know that enzymes are all mostly synthesized by the animals, but vitamins are not synthesized by animals, so they have to be taken in our diet. Vitamin D is either supplied in food or produced in the skin by irradiation of sterols by UV light. So you must be knowing that nowadays many people are suffering from vitamin D deficiency, which causing a lot of pain in our body, especially the bones. So the doctors are suggesting that we should be exposed to sunlight minimum 1 to 2 hours a day. And you might be seeing that people, usually the foreigners, they spend a lot of time in sea beaches just by exposing their body in the sunlight. So it really helps in synthesizing vitamin D in our body. Human being can synthesize vitamin A from carotene. So when we take carrots, vitamin A can be synthesized within our body. So vitamin A, as you know, is very important for our eyes. So it is important to incorporate carrot in our diet. They are chemically different from the main nutrients such as fats, carbohydrates, and proteins. The absence of or deficiency of a vitamin can cause a specific disease. So you know the list of vitamin deficiency diseases are there in the textbook. Which the deficiency of which vitamin causes which disease you will find in the textbook. Multiple deficiencies caused by lack or more than one vitamin are common in human beings. The condition of vitamin deficiency is known as avitaminosis. Okay, please try to remember these terms. When you have multiple deficiencies of vitamins, it is called avitaminosis. A, as you know, most of the cases it is negative. Okay, no vitamin. So classification of vitamins also we have. Vitamins are classified as water soluble and water in fat soluble vitamins. Under the water soluble vitamins, we mainly have B group vitamins and vitamin C. A very commonly asked question is, why should we take vitamin C regularly in our diet? As you know, it is a common Indian tradition that when you are served lunch or dinner, mostly lunch, not dinner, you will be given salt, a green chili, and a piece of lemon. Okay, so that lemon is serving as the requirement for your vitamin C. It is a normal tradition. Even when you go for wedding parties and all, you will see in the salad group that lemon and all these things are kept. Now, vitamin C being soluble, it is excreted out from your body from time to time, and that is why it is very important to incorporate in your diet regularly. It's not that one day I'm taking and forgetting for rest of the days. And now, you know, in COVID time also, it is so much suggested that you should take vitamin C regularly. So you can see it's written, this vitamins must be supplied in diet because they are regularly excreted in urine and cannot be stored except vitamin B12 in our body. Rest of the vitamins are fat soluble vitamins. They are A, D, E, and A, D, E, and K type of vitamins. They are stored in liver and in adipose tissues. The group of hydrophobic lipid soluble vitamins are not absorbed in the body unless fat digestion and absorption process proceed normally. The deficiency may cause malabsorption, absorptive diseases. Excess intake of vitamin also causes a disease that is called hypervitaminosis. So please remember, lack of vitamin causes avitaminosis. Excess intake of vitamin causes hypervitaminosis. So there is a table in page 418 as per my textbook. You may check your textbook. Please go through the table. It's very important. May be asked in the exams. Questions from there may be asked in the exam. The last topic of this chapter is nucleic acids. So you know there are two types of nucleic acid in our body. One is RNA, another is DNA. Now what chemically these nucleic acids are? Nucleic acids are also polymers. Okay, they are polymers of nucleotides. Alright, so let us see. All living cells contain nucleoproteins, that is a substance made up of proteins with biopolymers known as nucleic acids. Nucleic acids are long chain polymers which are present in high concentration in the nucleus of the cells. They play an important role in the development and reproduction of all forms of life. They direct the synthesis of proteins and are responsible for the transfer of genetic information, that is the hereditary characteristics. The repeating units of nucleic acid, Na stands for nucleic acid here, is called nucleotides. Okay, so nucleotides, when they undergo polymerization, they give rise to nucleic acids. Thus, the nucleic acids are also regarded as polynucleotides. While proteins have a polyamide chain, nucleic acids have a polypeptide ester chain. So please try to remember the names of these linkages. Okay, in carbohydrates we have a glycosidic linkage. In proteins we have peptide linkage, and in nucleic acids we have polypeptide ester linkage. Okay, now the two nucleic acids are DNA and RNA. Full form of DNA, as all of you know, deoxyribonucleic acids, and RNA is ribonucleic acids. That means the two nucleic acids, they differ by just one oxygen in the molecular formula, and of course they have difference in sugar units. Sorry, nitrogenous bases. So when DNA and RNA, they are hydrolyzed, the basis of hydrolysis, you know, we hydrolyze substances to know what is the monomers present in them, or what are the major constituents present in them. So upon hydrolysis, this DNA and RNA is found to give you three components. One is a nitrogenous base, heterogeneous base, and a 5-carbon sugar and a phosphate group. Okay, the 5-carbon sugar in DNA is deoxyribose sugar, and in RNA it is ribose sugar. All right, phosphate is common in both of them. Then what is nitrogenous base here? Nitrogenous bases are of two types. One is called purine ring, another is called pyrimidine ring. Again, I would suggest you to refer to the textbook. There are purine rings. Purines are two rings fused together with nitrogen as the members, and pyrimidines are usually 6-membered rings with nitrogen as their members with different attachments. Okay, now adenine, guanine, cytosine, thymine, uracil, these are the different nitrogenous bases. Now in DNA you have ATGC pairing, that means adenine ATGC, and in RNA you have, instead of thymine you have uracil. That is very important and very often asked in the exam. What is the difference between DNA and RNA in terms of nitrogenous bases? Heterocycles present in nucleic acids are substituted forms of this compound. Complete hydrolysis of DNA and RNA yield a pentose sugar. So ribose is a 5-membered ring sugar, so that's why we call it pentose sugar. Okay, these sugars are beta D ribose. Beta D ribose, you understand, when they undergo cyclization, just like in glucose and fructose, the OH is forming on the left in the carbonyl group, and beta D2 deoxyribose in DNA. The phosphate group, these are responsible for linkage of the nucleic acid polymers. The phosphate group is bonded to a hydroxyl group of sugar. So the phosphate is actually, as you can see in the schematic diagram here, the phosphate is serving as the link between two nucleotide units. Okay, now what are nucleosides? Nucleosides are the molecules in which one of the nitrogenous base is bonded with the sugar molecule is called nucleosides. That means This schematic diagram, each vertical line represents a nucleoside, and when it is linked to the phosphate, it becomes nucleotides, and when they form the polymeric chain, they form the nucleic acids. Okay, so nucleosides are sugar unit and the nitrogenous base, and nucleotides are sugar unit, nitrogenous base, and the phosphate linkage. You can see here the nucleosides of RNA and DNA differ in two respects. The sugar in RNA is ribose, and DNA it is deoxyribose, actually 2-deoxyribose. That means from two positions the oxygen is removed. The nucleosides of RNA contain uracil base, and that of DNA contains thiamine base. Okay, so that is one very important difference. When the phosphate group is attached to the nucleoside, the compound formed is called nucleotide. Thus, a nucleotide is a phosphate ester of nucleoside and consists of purine or pyrimidine base and a 5-carbon sugar and one or more phosphate groups. So please check here the base, that is the nitrogenous base, 5-membered ring sugar, that is ribose or deoxyribose sugar, and the phosphate together make nucleotide. Now what does these lines, the schematic diagram mean? Here you see this is the sugar unit. Let me write again. This vertical line mean the sugar unit. Okay, and your nitrogenous bases are attached here in carbon 1, and carbon 3 there is a phosphate group. Now carbon 3 of one is linked to carbon 5 of the other nucleotide chain, and that's how the nucleic acids are formed. So the phosphate is linking carbon 3 of sugar with the carbon 5 of the other sugar only, but the other chain. So you know that DNA is having a double helical structure which is supported by um, base equivalence theory. That means when you hydrolyze DNA, the nitrogenous base which you get, as many number of adenine you get, exactly same number of thymine also you get. So it is understood that they always occur in pair, that is called ATGC pairing. This is another way you can represent the nucleic acids: sugar linked to base, then phosphate, and again sugar base. This way you can see the brackets is drawn by cutting the bonds, which indicates the polymer, but here the point of linkage is not mentioned. So according to me, the previous representation is better. In nucleotides, the sugars are the attachment of to the nitrogen atom of the heterocyclic ring by bond between C1 atom of sugar and nitrogen atom of the heterocyclic ring. The linkage is called N-glycosidic linkage. So what is this? What is N-glycosidic linkage? When the sugar is attached to the nitrogenous base, and you know in the nitrogenous base there is NH2 groups. So when the nitrogen is involved in the glycosidic linkage formation, it is called N-glycosidic linkage. Instead of oxygen here you have nitrogen. Otherwise the linkage looks similar. The phosphate group bonded to a hydroxyl group of sugar at C5. So phosphate group is attached to which group of the sugar? Hydroxyl group of sugar. As you know, ribose is also polyhydroxyaldehyde. And the consecutive joining of sugar unit of one nucleotide to the phosphate group of the next nucleotide results in a long chain polymer called nucleic acids. So in the nucleic acids also, just like protein, we have a primary and secondary structure. This information regarding the sequence of nucleotides in the chain of nucleic acids is called primary structure. So you can see here the sugar unit is same in all. The phosphate linkage is same in all. Now what is different here is the heterogeneous nitrogenous bases. So what are coming in the sequence? What are the nitrogenous bases here? That will make the primary structure. Okay, it is the sequence. Rest of the as the sugar unit. So what happens here? In the primary structure, the information regarding the sequence of nucleotides in the chain of nucleic acids is called primary structure. Nucleic acids have a secondary structure also. When a double strand helix structure in DNA, the two nucleic acid chains are wound about each other and held together by a hydrogen bonds between pair of bases. The two strands are complementary to each other because of hydrogen bonds are found between specific pair of bases, A with T, G with C, ATGC pairing. I think it's a well-known pairing. The double helical structure explains the base equivalence as well as the other properties of DNA, especially the duplication in human organism. You know that DNA is responsible for the hereditary transformation from one generation to the other, or from mother to the child, the parents to the child. The secondary structure, in secondary structure of RNA, helices are present which are single stranded. Okay, sometimes they fold back on themselves to form double helix. RNA are of three types. They are messenger RNA, ribosomal RNA and transfer RNA. Now the difference between DNA and RNA, I request you to make a note on that. Please try to make a difference consisting of at least 3 to 4 points. So with this we come to the end of this chapter. Thank you. If you have any doubts, you can always ask.